Good morning, everyone. Hope you're enjoying PepTalk so far. My name is Prem Mohanty. I am a life sciences product specialist and product marketing manager at Benchling. I'm excited to talk about cell line development and some of the critical considerations in the associated data management to really accelerate your R&D. As you all know, cell line development is a process of generating large populations of identical cells that are well characterized. These cell lines are used widely across life sciences R&D. Some of the most common applications include production of antibodies and proteins, drug screening in discovery, in vitro testing, especially to study efficacy, safety, and tolerability, and finally, for fundamental research use such as functional genomics and study of cellular pathways and targets. Based on our experience working with customers doing cell line development, we see three major trends in the field. First, novel approaches to optimizing codons, constructing vectors such as plasmids, and engineering and selection of host systems are of active research interest. Second, the cell line screening strategies and processes continue to be optimized and automated to improve process flexibility, cell line stability and titer, and increase robustness in producing high quality cell line. And finally, comprehensive characterization continues to be an area of focus to ensure phenotypic and genotypic stability of cells throughout in vitro aging. An increasingly diverse set of orthogonal characterization assays are being used. If we look specifically at typical cell line development teams, we see several common challenges. A cell line screening scientist is typically using different software tools for managing incoming requests, performing in silico molecular biology and screening, which require manual effort to transfer and link data across the systems. A cell line analytical and testing scientist is typically running a suite of assays. Aggregating and analyzing these assays is a challenge. For a cell banking scientist, the software tools used to track inventory is disjointed from the ELN that contains the experimental context. This makes lineage tracking cumbersome. We have seen our customers align their efforts into scientific platforms. So it's not just focused on a single product, but instead they focus on a distinct pathway or technology capable of producing multiple therapies across multiple indications. As the science becomes more complex, we are seeing teams become much more specialized to increase the depth and efficiency they can bring to each step in the R&D process. Each of these teams is producing not just more data, but more complex data. There are more contextualizations that need to be maintained as you move downstream in the process and across programs. There are more streams of data, from lab automation and manufacturing sensors to newer sources such as real-world data. So the industry is looking towards digital solutions here to not just manage the volumes of data, but to bring order and structure to it. We think every company and scientific team deserves a solution that will accelerate and support your innovations. When you think about the ideal traits that any digital solution should have in R&D, there are four core attributes to consider. First, it has to model the science you focus on, which means having the ability to build data models that fit your science. Second, it has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. Third, it has to empower users with data at every level, giving scientists insights about their experiments and team leaders data on their programs so you can make both scientific and operational decisions quickly with complete and accurate data. Finally, it ensures that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will help keep them compliant and consistent with the rest of the R&D organization. Let's look at each of these attributes a little more closely. So what does it mean to be built for complex science? There has to be alignment between the experimental workflow and the way that the underlying biological and chemical entities are modeled. As an example, in the case of cell line development, the science here generally involves the iteration of vector design to express and purify novel proteins. screening and cloning to select ideal clones, characterization of clones, and cell banking. While this is a very simplified view of the process, it's actually very complex to model this. Every cell line has a variety of entities associated with them, such as genetic markers, plasmids or viruses used for the modification of the cells, lots prepared, passages, etc. Each of these entities have their own sequences, properties, and ontology. Every resulting cell line has experimental results that need to be tied back to not just the clonal hits, but also its underlying parts and sequences. So this is clearly a data modeling challenge, but it's also an operational challenge, since these also represent the day-to-day functions for lab scientists developing these cell lines. When we think of preparing digital infrastructure for these kinds of challenges, we have to think about both the data management and structure as well as the operational considerations. The way that we think about this is to build applications that follow the operational workflows from in silico design to experimental execution, but to make sure the data capture flows into a structured data model. This means at any point in the process, you should be able to trace your work back to all the preceding steps and underlying sequences. And more importantly, for scientific decision support, the data can be readily queried across any dimension of the data model. This is a necessity to start building in automation and machine learning routines. So that's an example of how a very complex scientific process can be structured into a digital solution. But the hallmark of any good program is that the science will evolve over time. So what happens when things change? Digital solutions have to be adaptable. The IT world has moved away from monolith software and towards more agile approaches. But adaptable shouldn't always have to mean coding and application development. Many life science companies either don't have the resources to do this or want to use the resources they have to focus on higher value operations. This is an illustrative example of part of a cell line development process, and it reflects the constant iteration and evolution that these specialized teams see on a daily basis. So, in an ideal world, the data model, the process, and the workflow should be configurable in a low to no code environment. So, companies can iterate and evolve quickly without being resource intensive or overly dependent on their digital partners. One way we seek to achieve One way we seek to achieve this is with a configurable data model that is editable right from an admin panel. So users can adjust the relationship and metadata on a particular entity quickly and without code. This also applies to process workflows, where you can update iterative processes like the example you see here. Once you have the data model and process in place to capture structured data, the next consideration is decision making. A modern digital solution should enable its users to make data driven decisions. Different members of an organization have different questions that they need to answer and have varying decision support needs. For a scientist, this means uncovering insights across their experiments so they can design, execute, interpret, and optimize their studies more efficiently without tedious data aggregation and data cleanup. For example, a scientist might be looking for an appropriate cell line to plan the study. Now, instead of emailing other team members or going to separate inventory and ELN tools to look for this information, the scientist should be able to look up and compare expression profiles and inventory data of multiple cell lines by querying a central data warehouse, where the data is already standardized during data capture. At the level of a team manager, the questions that need to be addressed are at the process and team productivity level. Now, typically this information is collated and visualized with Excel or other specialized process management tools with separate data entry and reporting workflows. A modern digital solution should be able to connect the experimental and process level information in a single system, such that managers can track team productivity and throughput easily and answer questions such as number of outstanding requests, assay turnaround time, and view the latest progress on related experiments. Finally, at the executive level, you want high-level view of your entire pipeline. the ability to compare candidates to make go-no-go decisions and allocate R&D resources efficiently. For example, the executives of a cell line development organization might want to look at key data across leading cell line lots to identify the best lot to support major development campaigns. Ease of use is a final important attribute of a modern digital solution. Most software tools have a learning curve, and then because of poor user experience, a forgetting curve. Only if the solution is easy to use can it ensure that users not only get trained but ramp usage and realize the full value of the solution. An easy to use solution allows users to record experiments quickly, tap into the records to uncover the most meaningful results, and finally aggregate and analyze results across studies from the same UI. This is essential to drive performance, boost productivity, and improve data integrity. We are proud to work with several leading biopharma companies that have helped develop new advances in medicine. We are closely aligned with these leading innovators and work in partnership. This has helped build up our domain expertise in biopharma and cell line development more specifically. So, how can we help cell line development teams? Looking across a typical cell line development workflow, from vector design to clonal selection and banking, we can provide a complete end-to-end solution for the entire workflow. Starting with cell line screening scientists, we can help integrate in silico molecular biology design with experimentation so that they can screen clones more efficiently. For cell line analytical scientists, we can help capture and analyze characterization data across assays to help with optimal clone selection. Finally, for cell banking scientists, we can not only provide a complete real-time view of the inventory, but more importantly, provide ready access to the full lineage and experimental history of the cell lines in the inventory. Looking a little bit deeper, there are three ways in which we can help cell line screening scientists. First, with our request application, we can ensure that there is a smooth handoff of all relevant information with an incoming request. So for example, with an incoming transfection request, we can ensure that the complete information such as parent cell lines, plasmid sequences, and experimental protocols are received. This prevents unnecessary back and forth communication between teams, and a scientist can thus start their transfection experiment promptly. Second, with our molecular biology application, you get access to a comprehensive suite of the latest in silico tools to design and optimize transfection vectors in a high throughput manner. This cuts down the need to use a variety of different molecular biology tools, reduces manual effort, and boosts throughput to meet screening objectives. Finally, our notebook templates and results tables allow you to centralize screening data on pools or hits and automatically links results to these entities. This provides easy access to all relevant results in one place for you to view and make decisions. We can help analytical scientists in these three ways. First, we can integrate with analytical instruments and lab automation to help capture results directly from these instruments into our notebook and automatically link these results to hits in the registry. This improves data integrity and reduces manual effort in transferring data between the software tools. Second, our Insights application, you can create complex dashboards to analyze characterization data of hits side by side. This simplifies data aggregation and analysis across studies and programs. Third, you can use custom dashboards to get a real-time view of the testing performed by your team. This helps managers improve productivity and optimize resource allocation and utilization. With cell banking, we can help you effortlessly trace sequence-based information and cell modification history of master cell banks and working cell banks. As most of you know, this is not possible with most software solutions today and requires heavy lifting from scientists to uncover this information across teams and software tools. Because of how interconnected everything is on Benchling, we make the process of lineage tracking easy. With just a few clicks, you can confirm the parent cell line, DNA markers, and the full experimental history of a cell line. Second, we can help you execute passaging experiments quickly and efficiently. Our templatized notebook entries allow scientists to not only set up experiments quickly, but also capture data in a structured and complete manner. Finally, we can help you manage your inventory of master cell banks, working cell banks, and other related samples with visual dashboards. This provides a real time view of your inventory and helps with study planning and inventory allocation to support critical studies. Everything I have talked about just now is powered by the Benchling Life Sciences R&D Cloud, which is designed to give you a complete view of your R&D innovation. Benchling's applications span the R&D process from experimental design, in silico biology, sample tracking to process management. There are 7 applications that Benchling offers, but they are all offered from a single interface. This makes it easy for users to leverage what they need without leaving the software. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. Benchling is built on a modern cloud infrastructure fully managed by Benchling and offers state of the art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that their R&D depends on. So we provide an open platform with full set of REST APIs and out-of-the-box connectors for lab automation, as well as ways to move data onto and out of the platform. I can share an example of how Benchling supports the cell line development teams of a large publicly traded biotech. This customer of ours is a leader in antibody based therapeutics based on the East Coast. Prior to Benchling, they used a variety of point solutions and custom software built in house. This made it difficult to keep track of important information across these multiple solutions. The second challenge was that there was no mechanism to enforce standardization and data capture, which meant that it was difficult to make sense of the data sometimes. And the third challenge was that bulk of the processes were dependent on legacy software and paper-based records that were no longer sustainable and had reached end of life. They adopted the full benching platform back in 2016. Their entire cell line development organization can now collaborate better around a single unified platform with a complete view of their experiments, samples, and results. We integrated our platform with some of the custom solutions that they wanted to keep and replaced the rest with benchling applications. We created standardized processes and templates for their teams to perform their experiments and register cell lines. We streamlined their entire workflow end to end on benchling, increased throughput, and improved visibility of data and processes across the organization. This is just one of the many examples on how Benchling customers can accelerate their R&D using Benchling. And you can find many more of these success stories on our website. Benchling's mission is to accelerate life science innovation for the betterment of humanity. We have built out a range of solutions for life sciences companies on a single platform and continue to invest aggressively in our product development teams to bring even more powerful and transformative features and products for our customers. Please reach out to me if you have any questions or would like to learn more about how we can support you and your teams. Thank you for joining me today and for submitting questions. Have a good day.